I'd like to give something back other than tissue.
Tissue is valuable. Well, that is
That's a
valuable. That's a very
I get it.
generous gift.
I feel like we had to have probably worked on yourself
I think it's
before.
great.
Just
So
have
I've
no
been
idea.
here before. So this is like the
You may
still be.
20th century molecular
Well,
version
yeah.
of.
somewhere.
You'll be happy
Deja
to know
vu.
that HIPAA stands very
Well, I can give you my
regulated.
number.
So if you,
You
have
were
it-
anonymized with a number, right? So my cell probably had a number to it.
Yeah,
And I think I asked Catherine to give me that number. They did.
we know everybody by their numbers, and that's it. So,
I'm sure I've been here before. I feel like I've been here before.
like a part of you has visited.
Yeah, yeah. I think it's a glitch in the matrix. Wow. Woo.
It's okay. So here's the Myoblasts, FSHD.
And so what bases are they in with that liquid?
So these are myoblasts.
So myoblasts are
Myoblasts
after
are
the
like-
stem cells, but precursors to myotubes, right?
Yes.
Correct.
Right. This is fascinating. It's one thing to talk about it, it's another to see it. You know?
You're gonna do another intro that this is the lab and we're back here doing, I don't know.
Yeah, you're recording now, isn't it?
Yeah, I am.
Okay, well there's the intro. We're back in the lab. In case you didn't.
Lab stuff.
Yeah, look at this. This looks like an advanced taser gun or something.
That is a pipette.
Oh, these are no longer blasts.
No
So
kidding.
yeah, we can put a
These are tubes.
pipette in there to suck up liquid.
Oh, okay, yeah. So
And
these
are micro
Robert.
pipettes.
correction. These are no longer myoblasts. He's differentiating them into myotubes. So these are actually myotubes.
Wow.
So
Now,
give me
can
one
you see
second?
DUX4 impacting the ability or during cell differentiation? Is that in vitro?
we can, but we have to basically freeze the cells. So we would have to stop them growing, fix them, so kind of just essentially freeze them in the state that they're in, and then we could stain them, or we can look for DUX4 molecularly, so we could, lyse the cells and remove the protein and look at it that way, or look at the RNA and look at it through qPCR.
So my project here is to differentiate them and reach the myotubes, and then I'll collect the cells, collect the RNA for the DUX4, and try to quantify or detect the DUX4 of these cells with other micro RNAs.
Oh, now this is
These
living.
RNAs working.
Yeah, so
Oh, well, it's
essentially,
live, actually. I mean, but
essentially what I've done here is, so you're seeing what I can see through the microscope. So we've just changed it. This knob here takes the view and the light path from the eyepiece, so what I can see, to the camera. So this is just like essentially a bright field image. So, the camera is a little slower than what you're doing, but these are the tubes
wow.
forming.
Now, in the context of an existing muscle cell or myofiber.
So that's essentially what this is. These are myofibers. So when we take them, when Mustafa cultures the myoblasts,
First, at the beginning.
we have to use certain conditions when we culture them. So they have different growth factors and different things in the media, which is the pink stuff that feeds the cells. So there's certain factors that help keep them in that blast state, or when he switches it over to differentiation, certain features of the media that gives it the signals to differentiate.
So you could keep them in the myoblast state for as long as you care within reason?
Yeah, I mean, essentially there's going to be a point where they can't stay that way anymore,
Right.
but these,
Wow.
some cells are immortalized, and that means that, you can
Definitely.
passage them for a very long time.
Very cool.
So proximity of the cells also matters. So when myoblasts, if they get too close together, they'll start to talk, and they may start fusing even if they don't have the features. So it's all in the way that you culture it, all about the factors that you give them to try and mimic what would be going on in the natural state on the plate.
And if I may ask, if you're looking at DUX4 production or induced, what have you, can you see, does it show that these disintegrate, or they don't form at all, or you're not looking at
So
it that way?
most of the time where you're going to see DUX4, yeah, see if Hossein can pull up his RNA scope. So Hossein has been working on a method that he can look in the cells. It's an in situ hybridization method.
What can I ask what that means?
So that basically means that he's essentially staining the RNA. And so he can find where that DUX4 message is being expressed. So we know that DUX4 is produced in the nuclei, which it's a little bit hard to see here. I can see if I can. So Mustafa is culturing these myotubes.
Differentiating the myoblasts.
Are you looking for
Bruce
biomarkers?
has some RNA scope stained.
Different biomarkers, microRNA, and DUX4.
I
So
see.
we're looking at the molecular profile to see if we can translate it into either readouts for clinical trials,
Right.
or if we can see the same kind of things in tissues or perhaps muscle biopsies or something like that. We're basically looking for readouts that we can use for clinical trials.
And you're looking at this at the time of muscle differentiation, which is what this is.
Well, we can take them at all different time points. So we can look at them at the myoblast stage. We can look at them right at, fusion. We can look at them after they've been differentiated for a while. We could treat them with, say, the microRNA that we're using as a therapy and see how that affects the biomarkers.
Can you look in as early as stem cell type deals, or not?
I mean, we can look as early as what the cells come to us as.
Okay.
So these are all myoblasts. So as you were saying, they're not true stem cells. They're already on a muscle pathway, but they're as stem cell as a muscle cell gets.
So here, like for
terms of
instance,
fashion.
one of the natural microRNA that Nizar mentioned he's working on, now we are trying all, as that was like on FSHD1, Myoblasts. And here now we try to find the correlation between the natural microRNAs that he's working on with the DUX4 and FSHD2 type of cells.
Yes,
So-
right. That's on 10
So
So
chromosome,
these
right?
cells in particular
yeah.
are FSHD2 cells.
I see.
Okay.
So
they're
this
from
is
an
a normal
FSHD2
D4Z4
patients,
repeat.
But a methylation.
Yeah, on
Yeah.
methylations,
Difference.
exactly. You're right.
So we're trying to see, because when we do these clinical trials, is it applicable to the entire patient population? Is it going to only work for part of the population? These are all things that we have to know,
Right.
for various
And I think I
reasons.
recently read that there actually may be an unknown other mechanism that's not been categorized yet.
Yeah,
There's
that's like
vague
there's some
thing.
patients they don't fit with the
102.
molecular. 102. They're still figuring out trying to figure out
Complex
what's
biology.
Yeah.
how it's
Good thing there's a lot of smart people working on it.
And they're right
Can I
here.
take the cells
They're
back?
right here.
They're all over.
Can I take the cells back?
Yes,
Because, I mean,
yes.
those
Sorry,
are
Can I take
we-
one to go?
That's like my babies.
I
I really
Can you bring
Don't
this to
touch my
my
hands.
I have
Yeah, no, no.
spent like literally on this project just growing them almost 1 month.
Wow.
Like yeah,
Get 'em
trying.
back
So
in
I
the
need to get them back to their incubator.
Very
Where
cool.
they're
You
where
want-
they're happy.
Thank you.
Yeah.
And I thought making beer was hard.
So Bruce can now show you this RNA scope image. So they are.
Oh, no, these are the
can see
nuclei,
here.
right?
Yeah, so it's just easier to see. So like I was talking about freezing the cells in a state, so these have been fixed and they're on a slide now.
Yeah, they are FSHD derived patients' myotube cells. They were actually myoblasts derived from some patient that we differentiate those in vitro to the myotubes. And after that, we try to looking for DUX4 messenger RNA with some custom designed probe and primers that we have for this messenger RNA. So they aren't protein or something. As you see, we were able to detect this
Wow.
signal in both nuclei and cytoplasm. Here, it's around 4 or 5 days after differentiation, and you can see the brown signal around the nuclei in differentiated tubes.
And that's what you put
So
in and
this-
introduced your...
This is DUX4.
No, that's
So the brown is showing where the DUX4 is.
Ah,
You can see that
This is
clearer
natural
there.
DUX4.
wow. So they always tend to
That's
conglomerate
endogenous.
together. They always come together, don't they? They're not just dispersed. They tend to come together, don't they?
So we can say that it's so normal thing that we could see, because the normal process from translating the messenger RNA into protein can occur on ribosomal reticle.
Ribosomes.
Yeah, and they are in the cytoplasm around the nuclei. So you can see those here. They are actually in the same place, because in the ribosome translation occur in that places.
Now, may I ask, this light blue, is this the myotube?
Yes.
So this is here.
Mm-hmm.
This would be, so it's
Yeah.
right here.
Yeah.
Let me get the way there.
So if we go to the another slide that it was for 7 days differentiated, we can see the signal even in the nuclei, in both places, nuclei and cytoplasm.
But what you can see in this picture is that not every
Yeah.
nucleus and not every cell is expressing the DUX4 messenger RNA. So it's in this culture it's sporadic.
And this is an FSHD.
One.
These could be your
Affected?
cells. We don't know.
Oh, okay. But this is not normal healthy tissue. This is
This is
diseased
an F-
tissue.
Yeah, these were cells derived from an FSHD.
I
patient
recommend.
biopsy.
I recognize that. Kilroy was here. That's mine.
Probably.
Wow.
And as you see, they are huge tubes that it's disformed tubes that we have in FSHD patients, derived cells.
And as you go on with differentiation, so this is day 7.
Day
What happens
5.
at day
At day
14 and 30?
At day 7, we will see bigger, larger tubes, but after that, they start to dying because accumulation of DUX4 in the cells is super toxic for cells. And that 7 days, we have huge amount of the DUX4 messenger RNA and protein. I can show you.
Oh, here's a
The
question
7
I've always
days.
wanted to know with DUX4. Is it itself, I understand the outcome is toxicity, right? But is DUX4 itself the toxic mechanism, or is it the body's immunoresponse that is the outcome that's,
I mean, it could be a combination of things, but I can tell you
When you
we've done an experiment years ago where so DUX4 is a protein that's called a transcription factor. And so the way a transcription factor works, it binds DNA
Mhm.
and then it turns on other genes. Okay? So we did an experiment years ago where we mutated the amino acids in that DUX4 protein that are required to allow it to bind DNA. But we still expressed lots of protein.
right?
Okay, the protein just wasn't capable of doing its normal function anymore. And when we injected that into a mouse muscle with the virus, it produced a lot of protein, but those muscles looked completely normal. So that would suggest that DUX4 has to actually do its job by activating
Right.
some genes.
And because
Now it's possible it activates genes that elicit an immune response.
Oh, wow.
But we know also these are
hey, that's a Canada goose right there.
Yeah.
It kind of looks like it.
Oh my goodness.
Yeah, I don't think
Where did he come from?
the cells in these
Look at that.
dishes, in these cultures. There is no immune system either. So
Ah,
when
so
it's
it can't be
killing
an
it, when it's
immune
killing
response.
it here, it's not because it's an immune response.
Yeah.
It's because it's something in that dish that's going on.
And this is
with
human
those
tissue,
cells.
so
That's
this
human
is-
tissue.
Well, then that's interesting then.
So
Very
at the
interesting.
7th day of differentiation, you see that fibers start dying. The form of the tubes changed from the previous one that you saw.
Yes.
And you can see more DUX4 in the tubes.
And the
amount of
DUX4 is the blue?
increased. No, the blue ones are nuclei,
I see.
and the brown is DUX for messenger RNA. That you can see that in some of the nuclei if you zoom that with the 100x objective, and in the cytoplasm. I can go even
Now, does this get to your,
closer.
what I heard you say, poly-A signaling that is protecting the DUX4 from dissipating?
Degradation.
Sort of that signaling component at the end of the that last deletion?
Yeah, the poly-A signal is required to stabilize a messenger RNA. If a messenger RNA doesn't have a polyase signal, it will, degrade quickly.
Yeah.
So for us to be able to detect these using this method,
Look at this.
we assume, but we don't know, we assume
Oh,
that
wow.
there must be a polyadenylation event added to the end of these transcripts.
Right. So it's really like a factor of three things. You have to have the unwinding relaxation.
Right?
Then you need, hypomethylation goes along with that. What are the other steps? And
You need to have the actual transcription occur.
right.
So something needs to bind the DNA and activate the gene to turn it on and copy it into a messenger RNA.
Okay.
That messenger RNA needs to be stabilized through polyadenylation. Once that occurs, then the messenger RNA goes to the ribosome and gets made into protein. Well, it serves as a template for the cell to make protein.
And I understand, is DUX4, you call it a gene and a protein?
The protein is called the gene product.
Okay.
So the gene is what we think of a gene. It's the DNA code of a certain, in this case, protein. Okay, so there's the DUX4 gene. That's the DNA.
Mm-hmm.
That DNA, that gene is copied into a messenger RNA, and that messenger RNA then serves as a template for the cell to make protein. So the protein is the gene product, and they're all called DUX4.
Okay.
Yeah.
It's just
But that's
different
just like the
stages of its
lifespan
life.
of a
That's
DUX4.
right. So, and when you refer to them, like there's nomenclature rules, right? So if you're talking about a gene and you write DUX4, you're supposed to italicize it. DUX4 italicized in a sentence
Right.
means the gene. And if you write
I see.
DUX4 non-italicized, it's the protein. So
you go.
it's subtle, but that's the convention.
Yeah, and actually detecting DUX4 in each level from DNA, RNA, or protein was so challenging and was so difficult till now. But this method helped us to detecting DUX4 at a messenger RNA level, so easier.
Okay, so if DUX4 is acting by binding DNA and then turning on some genes that are toxic, that's one idea for how it's operating. Yeah, if you could think about say a small molecule, like a drug, right?
Mhm.
That would be able to sit in that binding. Okay, so let's say this is DNA, right?
Right.
And DUX4 sits over top of DNA and binds it and then activates some genes. If you could find a drug that would sit in that binding pocket and prevent it, like
Yeah.
maybe close it up, prevent it from binding that DNA, you could think about that as a treatment. And there's some groups, both commercial and in academia, that are working on those kind of drug screens. The strategy we're using is not that. What we're trying to do is attack DUX4 at the messenger RNA level before it ever gets made into protein. The messenger RNA could be in different cell components depending on where you catch it. We hope we catch it early enough before it goes into the ribosome and starts being made into protein. So the idea would be to try to destroy it early.
And would this be a curative treatment, right? But this would never be passed down to offspring because you're still
Right.
going to have the genetic, but it would treat or cure the individual
Gene
throughout
therapy,
the rest of the life.
theoretically, should be a one-time treatment.
Wow.
But,
for something like muscle, where the healthy cell doesn't turn over a lot.
yeah, using the viruses we use are called adeno-associated viruses. They exist outside of the host cell nucleus, the DNA. They go into the nucleus, but they exist outside the host cell chromosome. So if that cell divides, when a cell divides, its DNA gets copied and passed down to the next cells, the daughter cells. The virus DNA that we're using
Mm-hmm.
does not get copied. It never replicates itself. And the cell, the host cell never can replicate that virus either. So if that cell happened to replicate or divide or die,
Mm-hmm.
the virus DNA would be lost. But like Lindsay said, in a condition like in muscle or nerve, cells that don't normally turn over very much, if at all, that virus should last, and so long as the cell is healthy, should last almost indefinitely, if not indefinitely. The reason I'm hedging on that a little
Yeah,
bit
yeah, yeah, yeah.
is because nobody's ever looked 50 years down the line with the gene therapy vector, right? The technology still isn't old enough to do that. But if it works the way we think it's supposed to work, if you deliver this to a person and it lands in the muscle and it helps to make that muscle healthy,
Mm-hmm.
it should be there theoretically forever.
Right.